Now that we have an overview of the existing standards and needs, Jackie Wolfrum will offer an example of an effort underway to fulfill one of those areas of need. Dr. Jacqueline Wolfrum is the Associate Director of the Biomanufacturing Program at MIT's Center for Biomedical Innovation, where she leads BioMAN's initiatives in the areas of cell and gene therapy manufacturing. BioMAN is an industry academic consortium focused on developing new knowledge, science, technologies, and strategies that advance the manufacture and global delivery of high-quality biopharmaceuticals. Jackie was part of the team that set up NIMBLE as well, the National Institute for Innovation in Manufacturing Biopharmaceuticals, which is the first NIST-funded innovation institute in the Manufacturing USA network. And she served as NIMBLE's Strategic Programs and Partnership Advisor for its first year of operation. Prior to MIT, she had more than 13 years of experience in assay and process development and validation and manufacturing technical support for cell therapy manufacturing operations as part of the Genzyme teams producing Carticel and Epicel, the first two autologous cell therapy products approved by the FDA. Prior to Genzyme, Dr. Wolfrum worked on research and development of a xenogeneic artificial pancreas device at W.R. Grace. Dr. Wolfrum received an SB in chemical engineering from MIT and her PhD in biomedical engineering from RPI. Please welcome Dr. Jackie Wolfrum. Jackie, the floor is yours.
So, thank you, Kendra, for your very kind introduction. It's my pleasure to be here this afternoon, and I will be discussing one of the projects that Don was referencing, which is SCB's efforts in standard development for rapid microbial test methods for sterility testing of regenerative medicine products. So a look at the autologous cell therapy manufacturing process shows why the need for rapid microbial test methods is particularly acute for these products. This is a schematic of Novartis's manufacturing process for Kymriah, which, as Dodd mentioned, was the first chimeric antigen receptor modified T-cell product to receive FDA approval. Autologous cell therapy products are personalized medicines that are manufactured from a patient's own cells and are intended for treatment of only that patient. The manufacturing process starts with collection of the patient tissue, which is then shipped to the manufacturing facility. In this case, the manufacturing process consists of isolation of the appropriate T-cell fraction, transduction with a vector containing the gene for the anti-CD19 receptor, and expansion of the transduced cells. The final product is a suspension of genetically modified T-cells, which is frozen and shipped back to the clinical site for administration to the patient. Autologous cell therapy products have a number of unique characteristics which have bearing on the sterility test, and I will highlight the most critical of these. First, living cells are the product, so the manufacturing process does not include viral clearance or terminal sterilization. Sterility assurance is obtained by validation of the aseptic manufacturing process under cGMP conditions and by sterility testing of the final product. Second, living cells have a very short shelf life, potentially on the order of hours to days. Cryopreservation can be used to extend the shelf life of the initial patient material and the final product. However, this will not always be feasible for all cell types and product configurations. Also, because of their complexity, it can be difficult to assess the impact of cryopreservation on performance of these products. Unlike other drugs, which are produced in large batches to treat multiple patients, a lot of an autologous cell therapy product treats only one patient. There can be significant patient-to-patient variability in cell growth, so there may be limited material available for sterility testing after the final product is prepared. A couple of other things to consider are that the manufacturing process includes the clinical site and extensive coordination is required. For example, CAR-T patients receive chemotherapy prior to treatment, so failure to successfully supply product on a scheduled date means that a sick patient has received an unnecessary procedure. There can be a relatively high risk of adventitious agent contamination because of the biological starting material, and also because the manufacturing processes are often highly manual and may include open operations. Finally, cell therapy products can have a variety of unique configurations ranging from single cells to cells combined with a medical device or full tissues. And we will see how several of these unique characteristics pose challenges for the compendial sterility test methods. So 21 CFR 610.12 provides the overall requirements for sterility testing of biologic products. FDA revised the regulations in 2012 in recognition of advances in the technology for detection of microbial contamination. The revised document provides manufacturers the flexibility to use new methods as they become available, as long as they meet certain criteria. The 2012 revision removed any references to a specific test method and instead provides general guidelines for both culture-based and non-culture-based methods. This approach, which is intended to encourage manufacturers to use the most appropriate and state-of-the-art test methods to promote innovation, address the challenges of sterility testing novel products like cell therapies, and potentially enhance sterility testing of existing products. The FDA requirements for the sterility test are relatively straightforward. First, the test must be appropriate to the sample being tested, such that the sample does not interfere with the test. Second, the test must be validated to demonstrate that it is capable of reliably and consistently detecting viable contaminating organisms. And third, the test and the test components must be verified to ensure that the test can consistently detect the viable contaminating organisms. So USP 71 is the compendial growth-based sterility test method. One important thing to note about the compendial method is that it is considered to be validated for its intended use. As Dawn described, the standard test method is an explicit set of directions of how to perform the test. This slide shows only some of the level of detail that is included in the compendial method. There are directions on how to perform the tests, how to test different types of samples, which growth media to use and how to prepare them, incubation times, the duration of the test, and how to interpret the results. The description also includes instructions for the sampling plan, which is based upon the final product configuration and the production batch size. It also specifies the standard organisms to use for demonstration of method suitability and growth promotion. You can see that there are several characteristics of this compendial method which make it problematic for use with autologous cell therapy products. First, it takes 14 days to get the final result. So for products with a short shelf life, this means that the product is administered to the patient prior to completion of the sterility test. Second, the readout is turbidity of the growth medium, which is problematic if the product being tested is a concentrated cell suspension, which is in itself causing turbidity. And finally, the sampling plan is not compatible with the case where the batch size is 1 unit and the amount of material available for testing may be minimal due to the need to return as many cells as possible to the patient. This slide summarizes the process that a manufacturer goes through when they implement a sterility test. The process for implementing the compendial method is shown on the left. The SOPs and material specifications are aligned with the relevant guidance documents. The only laboratory work required is method suitability to demonstrate that the sample configuration does not interfere with the test method. So in general, the process of implementing a compendial method is straightforward, relatively low risk, and relatively inexpensive. The process for implementing a non-compendial method is shown on the right. Manufacturers can use an alternative method, but they must demonstrate that the new method provides an assurance of safety that is greater than or equal to that of the standard method. Additionally, the alternative method must be validated. USP provides guidance on how to do the validation. However, not every new microbial detection technology will be capable of meeting the validation requirements. So in general, this route can be potentially complicated, high risk, and requires a significant investment in both time and money. I'd like to share with you the experience of Genzyme's validation of an alternative sterility test method for their Carticel product. Carticel is an autologous cultured chondrocytes for repair of cartilage defects in the knee, which was approved by FDA in 1997. The manufacturing process follows the general outline that we saw previously, where a patient tissue sample is shipped to the manufacturing facility for processing. The Carticel process consists of isolation of chondrocytes from the cartilage biopsy and expansion of the cells through up to 3 passages. The final product is a single-cell suspension which was shipped to the clinical site for surgical implantation. The Carticel final product has a shelf life of 72 hours, and there were up to 4 sterility tests performed for each lot. One at the end of the primary and secondary expansion cultures, a pre-release sample taken after the last feeding of the assembly culture, and a test on the final product itself. This table summarizes the test status at the time of final product lot release. Final results would be available for the primary and secondary expansion cultures. The pre-release sterility test would only be on day 4 of the 14-day incubation, so it was incomplete, and of course the sterility test of the final product would be incomplete. So what are the implications of having an incomplete final product sterility test at the time of lot release? First, there is increased risk for both the patient and the organization since final product sterility is not assured. Second, there is an increased amount of testing required to provide some measure of sterility assurance, and this alternate approach is summarized in FDA's 2008 guidance document on CMC for somatic cell therapy, INDs. And finally, the complexity of manufacturing operations is increased. Manufacturers will require SOPs for responding when a final product sterility test comes up positive after lot release. If this happens before the product has been administered to the patient, the shipment can be interdicted. If it happens after administration, then the patient must be monitored and any adverse event investigated to determine whether it is product-related. There will obviously be also an immediate and urgent failure investigation and CAPA, as well as increased reporting to the FDA. Because it is clearly better to intercept potentially contaminated materials before they get implanted into the patient, a sterility test which has a faster time to detection significantly reduces risk to both the patient and the organization. In the early 2000s, Genzyme selected the BACT-Alert system as having the potential for a more rapid sterility test. BACT-Alert, which was FDA approved for use for testing clinical samples, automates the direct inoculation growth-based sterility test. The media bottles have a colorimetric sensor on the bottom to detect CO2 evolution, which is correlated with microbial growth. The bottles are incubated in custom incubators which automatically scan the sensors on each bottle every 10 minutes. This figure shows the results from one of the validation tests demonstrating that the BACT-Alert system detected the presence of contaminating microorganisms more rapidly than the compendial method and often did so in under the 72 hours corresponding to the Carticel final product shelf life. FDA approved a 7-day sterility test using the BACT-Alert for Carticel manufacturing in 2004. The overall development and validation effort took 4 years and involved changes in the type of culture medium, incubation temperature, and incubation time. There are 2 things to note here. First, since the BACT-Alert is not a compendial method, every organization that wants to implement it would have to carry out the extensive method validation described here. And second, while implementation of the BACT-Alert was a significant achievement because it did reduce risk to the patient and the organization, a 7-day test is far from the ideal of having a completed final product sterility test at the time of lot release. As you are all aware, since you are here, there is widespread interest in rapid microbial test methods. The USP Modern Microbial Methods Expert Panel recently issued a stimuli to the revision process outlining the need for improved sterility test methods. Their report summarized the shortcomings of the current test as well as the most challenging user requirements that an improved test needs to meet. Time to detection was identified as a critical need. They also reviewed potentially useful analytical platforms and ended their report with the recommendation for collaborative development of a generic rapid sterility test. The standards coordinating body has undertaken that collaborative development project for the regenerative medicine space and is working on a consortium-based assessment of novel rapid microbial test methods. The goals of this project are identification of potential technologies, development and validation of the methods that have the appropriate performance characteristics, and training of the regenerative medicine workforce in use of these new methods. The output of the project will hopefully be consensus methods which can be brought forward by the standards development organizations to become compendial rapid microbial test methods. SEB's initial work has focused on identification of potential standard methods. Our review of the currently available commercial methods confirmed that while no single method meets all of the user requirements, there are some promising technology candidates. SEB is also focused on providing guidance for technology developers about what performance characteristics any new method must meet. We have started development of comprehensive user requirement specifications for a number of regenerative medicine product types. Also important is identification of the organisms that a new method must be able to detect. SEB is compiling a comprehensive list of relevant microbial organisms including compendial organisms, organisms from the published literature, and environmental organisms which were encountered by members of the SCB team in their facilities. Our hope is to develop a consensus panel of organisms which can be used to facilitate technology development and method validation. Once the test requirements have been defined, SCB will then facilitate collaborative round-robin testing of candidate methods meeting these criteria to determine which among them may ultimately meet the requirements for compendial methods. The SCB's project on rapid microbial test methods is a good example of the standards development process, and we have learned some useful lessons about the types of challenges that will be encountered. The first thing to realize is that standards development is a collaborative project. It requires the involvement of all of the stakeholders: therapy developers, technology providers, standards development organizations, regulators, and clinical groups. It is important to have clarity around the problem to be solved and what the needs are, and stakeholders should participate and contribute to the process to ensure that the standards being moved forward truly represent the consensus of the community. Second, standards are the culmination of a very lengthy process which benefits greatly from a staged approach. The steps involved include definition of the user requirements, identification of suitable technology, development of best practices, and all of the associated materials required for the compendial methods, such as detailed test methods, controls, and reference standards. It also requires extensive validation, comparison with the current methods, and uptake by the standards development organizations. Stakeholders should recognize that this is a multi-year process but can be broken down into a series of manageable goals, which allows flexibility in how stakeholders can contribute to the process. There are significant challenges which will be encountered along the way. One is that technology is constantly evolving, so a careful and thorough technology assessment is required to avoid prematurely committing to the wrong method. But also, stakeholders have to move forward even in the absence of a single gold standard technology. There is lack of precedent on how to do that, which introduces uncertainty. This makes it important to have input from a variety of stakeholders so that all viewpoints are considered. The regulated biomanufacturing environment is generally and appropriately conservative since patient safety is at stake. That being said, however, it is important to note that FDA is very supportive of efforts to appropriately introduce these improved methods because they do present a way to reduce risk to the patient compared to the current standard method. And finally, funding is critical to be able to execute all of the required activities for developing a new standard. So we would like to invite you to attend an upcoming workshop on rapid microbial test methods, which is being co-sponsored by SCB, NIST, and the two manufacturing innovation institutes, NIMBLE and BioFab USA. The participants will include representatives of all stakeholder groups. And so the goals of our workshop will be to review the current microbial test methods, regulatory guidances, standards, and reference materials landscape, identify the existing gaps, review a variety of analytical platforms that could be suitable to becoming rapid compendial test methods, to continue our work on collaborative development of detailed regenerative medicine specific user requirement specifications, and then we will also review technologies which are promising to fulfill the potential need in this space, and we will plan our next step forward. The workshop is being held on April 10th at NIST's National Cybersecurity Center of Excellence in Rockville, Maryland, and you can find out more information about the meeting on the SCB website or by emailing Allison Getz at SCB. So we hope to see you there. And with that, I'll say thank you for your attention this afternoon, and I'd be happy to answer any questions.
Jackie, thank you so much for that example, the lessons learned, and the invitation to the workshop. For those on the webinar, as a reminder, if you have any questions, please enter those now into the chat function by clicking on the question mark icon to the right of your screen. This may be a question mark or a circle with a question mark inside of it. Given the number of attendees in today's webinar, we'll be aggregating questions of a similar nature for the speakers or calling on specific questioners for the discussion. Jackie, the first question, you mentioned that uptake of standards like the one you're working on has been slow. Why do you think that is?
Well, I think one of the reasons is obviously that implementing the non-compendial standard is a lot of work, as we've seen from the Genzyme example. And so one of the things you have to have sufficient motivation in order to do that. And so, with typical biologic products, the standard compendial methods work well. And I think it's only really now that these regenerative medicine products with their unique characteristics, such as the short shelf life and so forth, are hitting the market, that there's really been a huge need for the new test methods for these products. We've seen that using standard methods is doable, but it involves a lot of risk and a lot of additional work. And so ideally, you would come up with new methods. Back when Genzyme was validating the bactealer, there were not that many companies that were involved in the regenerative medicine space. But obviously now with the pipelines that we have and the number of companies involved, there's much broader interest in having these methods available. And it would be a shame for every manufacturer to reinvent the wheel and go through the same process of validating a new method when the community could come together and collaboratively develop compendial methods, which would be much simpler to implement going forward.
No, thank you. Another question is, the CAR T therapy you discussed earlier managed to gain FDA approval last year. You mentioned several standards that do currently exist that were used in its development. Would the timeline or pathway to approval have been accelerated by the existence of any specific standards that don't exist today?
Let's see. Well, in general, I think that a lot of the unit operations that you use for cell therapy manufacturing are now trying to be validated according to standards. So I think Dawn had mentioned cell counting as being an important area where standards are needed. And NIST has a very extensive program working on standards for cell counting. But, in the absence of standards, manufacturers have to develop their own protocols and the justification for the approach that they're taking. And so if there were standards for cell counting, for example, compendial methods for implementation of rapid sterility tests, or compendial methods for measurement of vector integration into transduced cells, for example, the existence of the standards simplifies the processes that manufacturers have to go through, because the standards and the methods are accepted, and so there doesn't need to be as extensive a justification for why this approach is taken.
No, that makes a lot of sense. I think it could be helpful in general, it sounds like, across a number of areas. Also, is the rapid microbial test technology at a development phase where standardization is appropriate? And if so, kind of why or why not?
Well, I think what SCB has seen in their review, which is also what the USP panel also saw, is that there are a number of technologies which have advantage in certain applications. So there are PCR technologies which have rapid detection capability but require primers which recognize specific sequences. There are LED technologies which measure bioluminescence, for example, which are non-specific. And so while there isn't a single test which is encompassing all of the requirements, there are technologies which have advanced to validatable stages which encompass some of the requirements. And so one of the things that the community can do is come together and agree on a strategy for how do we move forward in the absence of a single universal test method. Is there a way to put together a panel of methods that have the necessary attributes and can be combined in a risk-based approach to achieve the goal, which is assurance of reliable and consistent detection of adventitious agent contamination?
Okay, thank you. And another question from the group. Do you think it is feasible to use different methods that are more appropriate for specific stages of the process, and then have a sponsor validate and adopt multiple methods?
Yeah, absolutely. And it may be that that's the approach that will have to be taken. right? Because as we mentioned, not all of the there's not a single method which encompasses all of the characteristics we need. And so in general, method development should be stage appropriate. And the idea with having this collaborative development project is that basically all of the manufacturers are in the same boat. And rather than individually trying to put together what that panel of methods might be and make the case that this is an approach that is suitable, development of consensus around that can provide a strong case for the regulators to see that this is, a community-wide investigation has come up with this approach as being the best way to move forward. And, that can enhance acceptance of the approach. And by collaborating on development of the approach and on the extensive development and validation of the methods which might be required, it's a way to share the burden so that every individual manufacturer doesn't have to do it on their own.
No, thank you. And you've implied a little bit, I think, as the answer to this question, but how do you think that in general we can accelerate uptake of standards for rapid microbial testing methodology or other regenerative medicine standards?
Well, I think there's been wide recognition that there's a need for these rapid microbial methods, and also recognition that it's a collaborative effort. And so really one of the things that's needed is someone to be the catalyst, to be the group around which people can come together and to kind of organize the effort. And so SCB has started that in the regenerative medicine landscape. Obviously, these methods are also of interest to manufacturers outside of this landscape, and SCB is, interfacing with the standards development organizations to work on a global acceptance of these materials. But I think there's a lot of activation energy involved, and so in the case of Genzyme, we had a very specific need at a very specific time and went ahead and did the development and validation on our own. But now that there's a community-wide recognition of the need and a lot more people who are now stakeholders, I think what was needed was the catalyst for bringing everybody together to work on the effort.
Okay. Thanks. And one, I think, final question, although they're kind of rushing in as we go here, is, regarding multiple methods, how practical do you think adoption of multiple methods is?
Well, I think that cell therapy and regenerative medicine manufacturers already see this in the cell characterization space because there's not a single method that characterizes a single product quality attribute that's indicative of product performance. And so, for example, with respect to potency, manufacturers are used to having a matrix of assays measuring a number of different characteristics that provide an overall evidence of potency. And so the situation may be no different in the sterility testing space. Since one method can't provide complete assurance in the time frame required, it may be necessary to have a matrix approach. But I think this is familiar to people who are already operating in the space, and so it should be generally accepted.
Okay. Well, thank you very much, Jackie, for, again, sharing kind of the discussion time and your answers as well as the overview. Very much appreciate it.